Okay, so let's talk about the nervous system. Our first introduction to the nervous system was when we were talking about the control mechanism for homeostasis. We talked about receptor control center and effector, and this is going to be our first control center here. You'll learn about the second control center. That's the endocrine system. That's in chapter 17, but that's in 211. So just to kind of go over some of the what fors and the general functions here of the nervous system, right? This is what, just think of it as a communications network. So this communications network is going to receive what we call sensory input from both the internal and external environment, and then it is going to process that information, and then it is going to determine some sort of outcome or effect. Right, and so it'll send that information on whatever the outcome or the effect is going to be through what we call motor output. We'll go through all this here. Right, so just kind of keep that in mind. It is our control system here. It's going to integrate all that information that I just talked about here, and then it's going to, as a result, through the effects of whatever the stimuli are going to regulate, right, certain functions in your body. Increase the temperature, decrease the temperature, right? Or cause shivering, not cause shivering, whatever examples you want to use. And so it does this all through electrical signal means, which we'll talk about nerve signals, action potentials, and whatnot. So if you kind of look up here, right, under functions here, collect information, processes and evaluate information, initiate response to information. This is just a more detailed way of what we talked about way back in chapter 1, stimulus, receptor, control center, effector. So what does the receptor do? It collects the information. Of what? Of stimuli, both internal or external. And then it's going to send that information through sensory input to the brain and spinal cord. And then what's going to happen there? Our control center is going to take that information and then process it and then figure out how it's going to respond. And once it determines how it's going to respond from whatever that sensory input information was, then it's going to then initiate that response to whatever that sensory input information was. And how's it going to do that? It's going to then regulate or stimulate an effector to carry out a specific response. So what's an effector? The basic definition of our effectors, we'll get into more detail, is muscles and glands. Those are effectors, muscles and glands. So you'll see, right, as we go through this course, how, and we'll go over several different examples of that. Right, so kind of keep that in mind. This whole spot here, the lower part of this slide, is basically just a review, but in a little bit more detail as to the homeostatic mechanism there. All right, so that's your nervous system, kind of like in a small nutshell there. A lot more to it, but in a small nutshell, this is what it's going to do. So we're going to talk about the nervous system now. We're going to break it down into the anatomical or the structural organization, and then we're going to talk about it in the functional or the physiological organization. So let's start off with the structural, the anatomy aspect. So our nervous system is made up of two parts structurally: the peripheral nervous system and the central nervous system. So what I suggest that you do, just memorize what's in the central nervous system, and then the peripheral nervous system is everything else. So your central nervous system is your brain and your spinal cord. That's it. Then the peripheral nervous system is going to be everything else. What does that mean? Well, it's going to be the nerves. So if you've heard of a named nerve like the median nerve, which is compromised in carpal tunnel, or what's the famous nerve that everyone always talks about? The biggest nerve, got pain down the leg, sciatic. Yeah, so sciatic nerve. So any named nerve is usually going to be part of the peripheral nervous system there. And so a nerve essentially is just a bundle of axons. And the other structure that you see up there is a ganglia. And the ganglia is a cluster of cell bodies. Now, we briefly went over a little bit of the anatomy of a neuron when we were studying the tissues back in chapter 5, but we didn't get into the actual specifics of a neuron, which we will in chapter 12 lab and lecture. So we'll get there. right now, just memorize that. Nerves in the peripheral nervous system are just a bundle of axons. And then a ganglia is a cluster or a group of cell bodies. And that's specific for the peripheral nervous system. Because guess what? Those two definitions that I just described to you, we give those structures different names in the central nervous system. We'll cover that right in a little bit. All right, so that's the structural part is the easy part. So central and peripheral. Functional, a little bit more to learn. And so the functional is going to have two components also, sensory and motor. So I mentioned before, the sensory is going to be the input information coming from the external and internal environments. That sensory information is going to come from those receptors and go to your control center, to your central nervous system. And so another name for sensory, for the sensory nervous system is the afferent with an A. And so essentially that's what it does. The sensory nervous system receives input, sensory input information from a stimuli, or actually, from receptors that were triggered by a stimuli. So there's two parts to your sensory nervous system. You've got somatic and visceral. So the somatic sensory nervous system is all the stuff that we can consciously perceive. So you can hear me right now. So you're utilizing your somatic nervous system. If you're seeing things, right, you're utilizing your sensory nervous system. If you can touch things, taste things, right? So your somatic sensory nervous system is going to include what we can consciously perceive. The visceral, on the other hand, think viscera, thinking organs, right? They're going to detect stimuli that you cannot consciously perceive. Like when your heartbeat goes from 55 beats a minute to 57 beats per minute, or when your respiration rate goes from 12 breaths per minute to 18 breaths per minute. You can't tell if your arterioles, which are some of your arteries there, okay, part of the arterial system, if they're 20% dilated or 40% dilated. You can't detect that, right? And so all of that, those visceral sensory, all right, system structures are going to be things that are just below, all right, our conscious perception there. So the other part to the functional, all right, organization of your nervous system is going to be the motor nervous system, also known as the efferent. And so now what we're going to see is on the other end of that whole kind of reflex apparatus. So I'm going to go back here. The other end of this. So now we're going to talk about, all right, with the motor, the control center has determined what type of response we're going to initiate from whatever the stimulus was. And so now we're going to initiate that response. And so our motor nervous system is going to initiate that motor output. Okay, it's going to get its marching orders from the central nervous system, and it's going to transmit that from the central nervous system out to the effectors, which are going to be muscles and or glands. So again, we have two parts to our motor nervous system. One is the somatic motor system. And so you remember from the somatic sensory system that it's things that you can consciously perceive. Well, the somatic is going to be the things that you can voluntarily control. And there's only one tissue type that you can voluntarily control. Remember, you have three types of muscle tissue, and then we have several different types of glands. I challenge any one of you to tell me how to control my glands, right? Tell me how to control my thyroid gland, because I need more thyroid hormone. I'm too tired. I don't need coffee, right? Give me some thyroid hormone, right? So when we're dealing with the somatic motor system here, the only tissue that you can voluntarily control is your skeletal muscle tissue. You can't voluntarily control cardiac. You can't voluntarily control smooth muscle. So the only tissue that falls under this is going to, under the somatic motor system, is going to be skeletal muscle. So everything else must fall under this one, the autonomic motor system. And so the autonomic motor system is going to send involuntary commands to the other tissue types, which is going to be smooth muscle, cardiac muscle, and your glands. And this, we'll see it later on in the autonomic motor system. There's two divisions to it: sympathetic and parasympathetic, fight or flight, rest and digest, that sort of thing. We'll talk about that in chapter 15. That's later. That's later. All right, so kind of keep in mind, a lot easier, right? Would you agree that the structural or the anatomy is much easier than this? Right, and this is where we're going to spend more time on learning here. But definitely know the differences when we're talking about organization to the nervous system here. All right, so this picture here is showing us that functional organization just in picture form. So you have the sensory nervous system, which is going to be bringing sensory input information to your central nervous system, brain and spinal cord. So the two types: somatic sensory, things that you can consciously perceive, hearing, smelling, touching, right, tasting. What am I missing? Smelling. I said smelling. Anyways, so that's your somatic sensory. Visceral sensory is going to be with mainly for your organs, all right? Blood vessels, stretch on organ walls and whatnot. Then your central nervous system will figure out how it's going to respond to whatever sensory input that it got from the stimulus, and then it's going to send its orders out to the effectors. So we call that motor output. And so we're going to utilize the motor nervous system, and there's two parts: somatic motor, which is what we have voluntary control over, which is only skeletal muscle. And then the autonomic motor is going to include all the remaining stuff of the effectors: smooth muscle, cardiac muscle, and glands. All right, so question for you. First question, well, not the first question of the night, because you just had 100 of those. The first question of lecture is going to be this: what are the two primary functional divisions of the nervous system? So what are the two primary functional now, divisions of the nervous system? We just saw them on the previous page. Yeah, sensory and motor. Good, good. All right, and then so how do they differ from one another? Right? So the sensory is going to do what? It's going to monitor stimulus, right? It's going to monitor a stimulus, both either external or internally. And it's going to transmit that information, whatever the stimulus was doing or did, to the central nervous system. And then the motor nervous system, that is then going to initiate and transmit through what we call motor output information, right? The commands from the central nervous system out to the effectors there. So that's essentially what we're looking at. Okay. So we're going to kind of switch back, take off our physiology hats, and put on our anatomy hats. And we're going to go over a couple of things here when we're talking about some of the structures here in the nervous system. So the first word that you see up there on the board is nerve. So we're going to be talking about those a lot. So it's good to get a good definition. So a nerve is a bundle of axons or parallel axons, bless you, in the peripheral nervous system. So make sure that you know that it has to be in the peripheral nervous system, because I can change 3 letters in that definition and it changes what that how we name a structure. So if I were to say a bundle of parallel axons in the central nervous system, we call that something completely different. We call that a tract, T-R-A-C-T. We'll learn this later on, all right? But for now, that's why I want you to understand that a nerve is a bundle of parallel axons in the peripheral nervous system, outside the brain, outside the spinal cord. So these nerves, as they're running through your body, down your arms, down your legs, right, they're going to have some coverings to them made of connective tissue. And there's 3 layers. And one of the things that you're going to learn here in this course I call it the rule of threes. There's a lot of things and structures that we'll be learning in this semester and in 211 that are associated with three things. And so for this, we're going to see that your nerve has three coverings to it. Right? You have the epineurium, the perineurium, and the endoneurium. So going from the outside and working our way in. Right? So the outside layer is called the epineurium. And this is made up of dense irregular connective tissue, which is good because dense irregular connective tissue gives it a little bit more support and structure. And it's a nice covering. So it kind of protects the nerve a little bit. So I'm going to show you a picture. I'll come right back to this slide. So here's a picture of the entire nerve here. So the whole outer covering to our nerve, that is going to be, right, the epineurium. Epi's on the outside. And then we're going to go to the middle, or you'll see that a nerve is made up of, I zoom in here, the nerve is made up of several smaller structures here. So you'll see there's one single nerve cell called a neuron. Right? And so the neuron is going to have its own individual covering. That's called endoneurium. So the endoneurium covers that individual neuron. It's made up of areolar connective tissue, which is a loose connective tissue. And then what we're going to do is we're going to take a bunch of our neurons, specifically the axons, and we're going to bundle them together, right, into a fascicle, which is just a bundling of several neurons. So we're going to bundle them up together and we're going to cover that up. And that's called the perineurium. Then we're going to take all the fascicles and bundle it up into the epineurium. All right, so that's what we're going to see here. All right, I should have started on the inside. Normally I do. I thought I'd try something a little bit different with you guys. Now I know why I do the inside first. All right, the endoneurium, that wraps each individual axon, which is part of an individual neuron, which is a nerve cell. And so the endoneurium is made up of areolar connective tissue. Now it's interesting because endoneurium is going to separate each neuron from its neighboring neuron. There's a reason for that. And the reason is listed right up here on the board. It electrically insulates each neuron. Neurons communicate and transmit information through electrical impulses. We'll learn all that. That action potential is there. So we can't have one neuron that's next to another interfering with the other neuron's function. Because some neurons are sensory and some neurons are motor. They can't be both, or one or the other. Right? So we don't want, while the sensory neuron is getting input information from a stimuli somewhere, we can't have it triggering a motor neuron to then affect some random effector, like the cardiac muscle. That'd be bad. All right? So we can't have that. So endoneurium, it electrically insulates each axon and separates those axons from each other. So we're not kind of crossing the streams or interfering with each other. So we're going to take a bunch of these individual axons. We're going to bundle them up and form these bundles of axons, and those are called fascicles. And we cover those fascicles up with a dense irregular connective tissue layer called perineurium. And then we're going to bundle up all the fascicles into what we call epineurium. And that's what we saw here. Right? There's your individual axon with its endoneurium, and then we'll bundle up a bunch of them into a fascicle, and then we'll cover that with perineurium. Then we'll cover the entire fascicles or the entire nerve with the epineurium there. Okay. One of the important things too, you'll notice it says up there that nerves are vascularized. Well, that's important because nerves have a high metabolic rate, which means they're going to require a lot of energy because they're going to be doing a lot of things. So we have to have them have a wonderful amount of blood. When I say wonderful amount, enough to sustain them. Okay, so they're going to be highly vascularized because we need to get a lot of nutrients and get rid of wastes, right, to our different nerve cells there. All right, so we'll talk about this a little bit more in lab, hopefully. And one of the things that you want to understand when we're looking at the anatomy of our nerves is where they're located. So again, another simple way to remember this. If a nerve comes off the brain, we call it a cranial nerve. If the nerve comes off the spinal cord, we call it a spinal nerve. Real basics. So you have 12 cranial nerves. Right, so you have 12 pairs because you have one that comes off the left and one that comes off the right. We'll learn about those later on in lab. So again, a cranial nerve is a nerve that comes off the brain. A spinal nerve is a nerve that comes off the spinal cord. So again, we're dealing with the structural or anatomical classification is much easier than the physiological. The physiological or the functional is a little bit more complicated. So if you look there up on the screen, you can see how when we're talking about nerves, mind you, you have sensory nerves, you have motor nerves, and then you have what's called mixed nerves. And the mixed nerves are the majority of the nerves in your body. But let's start off at the top there. Sensory nerves carry sensory input information from the receptors to the central nervous system. So they're going from the periphery to your brain and spinal cord. And that's all they do. They don't carry any motor information. Motor nerves are going to then, therefore, carry nerve information from your central nervous system out to the effectors. So they're essentially going to go away from the central nervous system. They originate in the central nervous system and go out to the effectors, the cardiac muscle, smooth muscle, or gland somewhere. And then mixed nerves, obviously mixed, you'll have both. So if you think of sensory and motor nerves as just one-way streets, okay, the nerve signals only go in one direction on both of those types of nerves. If you're sensory, you go from the receptor to the brain or spinal cord. And if you're motor, you go from the brain or spinal cord out to the effectors, one way. Mixed nerves, two-way highway. Okay, you go both ways there. But here's the thing, I mentioned this before. Even though, all right, with our mixed nerves, you have both sensory and motor information going in there, each individual axon, which is part of the neuron, which is the transmissible or transmitting nerve cell, it's either a sensory neuron or it's going to be a motor neuron. There's no such thing as a mixed neuron. It's either designed to carry sensory information only or motor information. But in your mixed nerves, you can have a mixture of these different types of neurons or axons there. Okay, and then finally, here's just a review, ganglion. Ganglion is the single, ganglia is the plural, right? But it is a ganglion is a cluster of neuron cell bodies in the peripheral nervous system. Again, keep in mind, peripheral nervous system. If I were to change 3 letters in that, if I were to say a cluster of neuron cell bodies in the central nervous system, we call that, talk about confusion, a nucleus. All right, how do you like that? You know, the nucleus is the control center for your cells, right? It has all the DNA and all that fun stuff. Well, I don't know whose idea it was to call it a nucleus for this, but we'll get there. We'll talk about it. Okay, so let's talk about some of the characteristics of, all right, our neurons. All right, so neurons have the following. They're excitable, they can conduct things, they're going to secrete stuff, they live a really long time, but unfortunately, they can't divide, except in extreme circumstances. I should be specific about that. But for the most part, your neurons do not divide. They do not undergo mitosis. So what is excitability? How a neuron responds to a stimulus. And we're going to talk about this term here throughout this chapter, but a stimulus can cause a change in what we call a cell's membrane potential. And we'll get into that later on in the chapter, but what a membrane potential is. It's essentially an electrical charge across the plasma membrane. We'll get there. All right, the next one up there is conductivity. Think about it, right? That word, pretty much most of us should know. How well, right, can a neuron conduct or propagate an electrical signal? And so we talked about, back in chapter 4, proteins. And one of the actually in chapter 2, we talked about it, but one of the types of proteins that we're going to talk a lot about in this chapter, what we call transport proteins. And we learned a little bit about that in chapter 4 when we were talking about how things move across the plasma membrane. Transport proteins move things either into the cell or out of the cell. So these specific transport proteins that we're going to learn about are called voltage-gated channels. Right, so a couple things in that 3-letter description there. Voltage means we're going to have an electrical charge of some sort involved. Then gated is going to mean that these channels have a gate on it. And that gate is either open or closed. All right, and we'll discuss all that. But I just want you to understand voltage-gated channels. And so we're going to see when these transport proteins are stimulated, they'll start to open up. One, then the next one, then the next one. It's like dominoes. If you push dominoes down into each other, they'll just fall sequentially. And so that's what we're going to see here, right? And we'll get into that. Secretion. At the end of our neuron is this structure called a synaptic bulb or end knob. And so it stores chemicals in there. We call these chemicals a neurotransmitter. And so the electrical signal that a nerve conducts will eventually stimulate the release of this neurotransmitter. And when it does that, we call that secretion. We'll get into those specifics there. All right, extreme longevity, pretty much these cells will last you a lifetime. Hopefully, if we're lucky enough. So you won't kill any of them unless you're drinking alcohol. All right. And then amitotic, which means they do not, all right, divide. And most of the neurons have lost that ability to undergo mitosis after, right, you have fully developed during your fetal development period there. There are exceptions, but I'll let you know when those are. Okay. Quickly, let's just kind of go over a quick review here for our neuron. So I'll show you a picture and I'll come right back to this slide, I promise you. Here's our neuron. So there's actually 3 of them. Here's part of one at the top of the screen. You got a full one here in the middle of the screen, and then you got the start of one at the bottom of the screen. But here's our neuron. So we're going to start at the body and then work our way down. All right. So the cell body, and that's that kind of expanded bulbous-like structure that you see, all right? We also call that the soma, S-O-M-A, which is also a muscle-relaxing drug, okay? All right? But the cell body is going to be where we're going to find our organelles, the majority of the organelles, like the endoplasmic reticulum, right? And a lot of the ribosomes there. The nucleus is going to also be found there, right? And so, of course, inside of our cell body, you've got cytoplasm, but we give it a special name. Okay, the cytoplasm inside of the cell body is called perikaryon. And it's here at the cell body that the neuron is going to receive stimulation, whether from another neuron or from a receptor or whatnot. And it's going to create these little impulses called graded potentials. They're little miniature kind of like electrical charges. Think of it like that for now. We'll get into detail. And so the dendrites, which are those processes that come off these guys here, these are the dendrites. And so the dendrites, right, their job is to receive sensory information or to receive some sort of electrical information from somewhere. Whoops, here we go. All right, what we'll see inside of the cell body is going to be our ribosomes that we can call either chromatophilic substance or Nissl bodies. And so those ribosomes, for whatever reason, I don't know why, I believe it was the person that discovered them. I'm assuming they were Dr. Nissl, all right? But The ribosomes, just like we learned back in chapter 4, are either free floating, they're free ribosomes, or they're attached onto the endoplasmic reticulum, and therefore it's known as the rough endoplasmic reticulum. So we'll see, all right, in a neuron, though, the ribosomes are either referred to as chromatophilic substance or Nissl bodies there. All right, dendrites are those short processes that I was telling you about before. One of the characteristics of dendrites is that they're unmyelinated. So what does that mean? It means they don't have a fatty covering on them. Okay, myelin is this fatty sheath that helps to transmit nerve signals faster. We'll get into it. Well, I'll discuss it, but you're going to just see the cell body, and this is normally where I try to draw this, and it falls apart. Right. The dendrites are these short processes that come off the cell body. Their job is to receive any input from wherever and then transmit it to the cell body. So it's going to transmit that information to the cell body. All right, so those are the dendrites. So off the cell body, you're going to see a really big process coming off. And so you would think, oh, that's just a big dendrite. No, it's not. It's actually called the axon. All right, so the axon is that long process. It's the largest one that comes off the cell body. And so this axon is going to travel down and is going to connect with one of these structures here, or more. Could be more. It could connect to, all right, another neuron. It could connect to an effector, which is a muscle or a gland. So those are your choices. Right? The axon is either going to contact a muscle, a gland, or another neuron. So right where the axon kind of comes off of the cell body, you have this kind of triangular-like area here. That would be this area right here that I'm circling. That's called the axon hillock. That's the beginning of the axon as it's attached onto the cell body. That's very important. Physiologically, it's very important because that's where we initiate what we call the action potential, the nerve signal, the actual electrical signal that is going to travel down the neuron or the axon. That is the location of it, originates here. Great test question to ask, right? The axon hillock is where you initiate an action potential. So in the axon, we're going to change names of some structures here that are specific to the axon, like the plasma membrane, right? The plasma membrane, we call that the axolemma. In a muscle cell, we'll call it the sarcolemma. So if you were talking about an axolemma, I would know that you were specifically discussing, right, the plasma membrane of an axon. Inside the actual axon, you have cytoplasm there, and so we give that a special name, and we call that axoplasm. So on some of our neurons, you will see on the axon, it will give off branches. Some neurons have a lot of branches. Some neurons have very few to none. So they're just these different branches that come off, and we call those axon collaterals. And so a nerve signal can come down the axon, it can go down this collateral, it can go down this collateral, and it can continue on its way down the main axon there. So those collaterals allow us to stimulate multiple, right, effectors or multiple neurons, right? Just a lot of different branches there coming off. So when we get to the very end of our neuron, we're going to see, whoops, hold on. At the end of our neuron, so as the axon's coming down, it's going to give off these smaller kind of processes, and those are called teledendra. And at the ends of these teledendra, we have these big kind of swellings at the end of them. And those are called the synaptic knobs. Right, so the axon just kind of gives off these little small branches coming off, and at the ends you have these swellings called the synaptic knobs, and in the synaptic knobs is where the neurotransmitter is. And the neurotransmitter is stored in these tiny bubble-like structures, balloons, whatever you want to call them. Those are the synaptic vesicles. And the synaptic vesicle houses the neurotransmitter. So an electrical signal will come down the axon. It'll travel down the axon, and then it will stimulate the synaptic knobs to release this neurotransmitter onto either another neuron that might be contacting this neuron, or it could be a muscle cell, or it could be a gland. All right. Quickly about the cytoskeleton, nothing really terribly exciting. You remember these different filaments here, the microfilaments, intermediate filaments, and whatnot. All right, well, the intermediate filaments, these neurofilaments here, are going to provide, all right, some support structure strength to the neuron. And actually, also, it's the cytoskeleton that plays a role. You'll learn in chapter 17 that certain hormones are made in your hypothalamus and are transmitted to your posterior pituitary utilizing neurons. And so they'll just transport in these vesicle structures from the body of the neuron in the hypothalamus, and it'll transmit it to the synaptic terminals there, the synaptic end knobs in the posterior pituitary. More on that later, but that's the only reason why I mentioned the cytoskeleton, because it can play a role there in other chapters. Okay, so here, just to kind of go over, let me zoom in a little bit. So, all right, so here's our neuron right there, and you can see another neuron at the top of the screen is attaching onto it. So here you can see, here's the axon giving off the telodendroid here. At the end of the telodendroid are these synaptic end bulbs or knobs. And these synaptic knobs, if we zoom in on them, you'll see they're full of the neurotransmitter stored in these little bubble-like structures called synaptic vesicles. So when that electrical signal comes down, it'll stimulate these synaptic vesicles to release, right, their neurotransmitter in this little gap area here, and that's called the synaptic cleft. And so those neurotransmitters will hang out in the synaptic cleft, and on the other side of the synaptic cleft, which is this second neuron, which we would call a post-ganglionic neuron, right? Or excuse me, post-synaptic neuron. It has some transport proteins here, some gates. That neurotransmitter will stimulate possibly the opening of gates or closing of the gates, and then something else will happen, which we'll talk about later on. But I just kind of wanted to throw that out there. Speaking of synapses, so we're now going to talk about what this is right here. Here's your synapse. You got your presynaptic neuron. That's this first neuron here at the top of the page. And then you've got your postsynaptic neuron. That's this big neuron in the middle of the page there. All right, so what is a neuron? It's a space. It's a location where a neuron can either attach onto another neuron or it can attach onto an effector, a muscle or a gland. So in our bodies, we have two types. We have chemical and electrical. So the chemical is much more common. We'll briefly talk about the electrical because we don't really spend too much time on the electrical. It's pretty much in the central nervous system and in your eyes, right? So in the brain and in your eyes. And the thing is, when you have a presynaptic neuron and a postsynaptic neuron that are attached to one another, they're attached through gap junctions. Do y'all remember? We saw gap junctions before in another tissue. Do y'all remember which tissue? Remember I'll tell you this right now. As soon as I say it, you'll know it. Intercalated discs. Yeah. So intercalated discs are those structures in cardiac muscle cells. And they're made up of desmosomes, which attach the cells to one another. They make it a nice snug fit. And then the gap junctions, which are these holes that allow cells to share things with one another. Okay, so stuff from one cell can flow directly into the second cell. All right, so here, when we're looking at the electrical synapse here, the nice thing about electrical synapses is there's no synaptic delay. None. And we don't have to convert our electrical signal into a chemical signal, then back into an electrical signal. Now, it doesn't mean much to you right now, but when we go through this chapter, you'll understand what I'm talking about. So here for electrical synapses, it's pretty much just straight up an electrical charge that can go from one neuron directly into the second neuron. So you're not going to see what we call a synaptic delay, because a synaptic delay is a slowing of the transmission process. And I'll explain that to you here. All right. In our chemical synapse, far more common here. So same type of scenario here. We're going to be dealing with two neurons. I shouldn't even say it. When you have a chemical synapse, it's either going to be another neuron. Our first neuron is either going to attach onto another neuron, or it's going to be an effector. But point being is, you have your presynaptic neuron. So the presynaptic neuron, the axon terminals, those telodendri, are going to be carrying the electrical action potential nerve signal down towards the synapse there. The other structure, we'll use neurons in our example, we have our postsynaptic neuron. So one produces the signal and transmits it. The other one, the second one, receives the signal. And usually it's going to receive the signal on the dendrites, right? But it can also receive the signal on the cell body. So again, I'll come right back to this slide here. That's what you're seeing here. See how this presynaptic neuron here, it has several telodendri. Some of the telodendri are synapsing here on this dendrite, some are on this dendrite, but some will go on to the cell body here. Again, keep in mind, this is what we're looking at for our chemical synapse. So you got the presynaptic neuron that's going to be producing the nerve signal. And so these synaptic vesicles are going to release the neurotransmitter here. Oh, golly, I keep forgetting that you're not seeing that. I see it on my screen, but you're not seeing it on your screen. All right, you're going to see these synaptic vesicles are going to release the neurotransmitter here into the cleft here. And then it's going to affect the postsynaptic neuron, and we'll talk about how it does that. All right, so three things that you have. Presynaptic neuron produces the signal, postsynaptic neuron receives the signal, and then there's a gap in between. It's important that you understand that. It's a fluid-filled gap, which means you're going to have the neurotransmitter diffusing across that fluid there. So this right here, 4 easy steps of how, all right, a synapse communication occurs. The presynaptic neuron is going to produce the signal through its action potential. Its electrical signal will come down the axon, stimulate the synaptic vesicles to release neurotransmitter into the synaptic cleft. So we're starting off with an electrical signal at first. And then that electrical signal stimulates that cell to release the neurotransmitter into the synaptic cleft. This is where things start to slow down here, right? That's why we have this term here called synaptic delay. So that neurotransmitter molecule is released from the presynaptic neuron. It diffuses across the synaptic gap there, all right, or the synaptic cleft. And then it's going to then bind onto the transport proteins, the voltage-gated, all right, channels there on the postsynaptic neuron. Well, that whole process takes time, and so that's going to slow things down slightly. So that's what we call the synaptic delay. So, question for y'all. What is the mode of transmission in a chemical synapse? Basically, the answer is all this stuff here. All right, just in a little bit of a different way of explaining it to you. All right, so molecules stored in synaptic vesicles are released from the synaptic knob of the first neuron, which is what we call the presynaptic neuron. So the molecules are released into the synaptic cleft. They diffuse across the cleft, and then they will bind onto the receptors on the postsynaptic neuron. And those receptors are attached onto voltage-gated channels of some sort. And all that stuff we'll get into. Because you're hearing me say voltage-gated channels and this and that, and some of you are probably like, I don't know what the heck he's talking about. No worries, because guess what? We're going to now start talking about some of these valued transport proteins here. So when we're talking about our neurons, you have two types of transport proteins, which we call pumps and channels. Okay, but regardless, there's both transport proteins. So here's our pumps, and it's just like if you have a basement at your house, you might have a sump pump in the basement. That sump pump pumps water out of your basement, so your basement doesn't get flooded. Well, guess what? The pumps here in your neurons are going to do the same thing. They're going to pump stuff out of the cell, while at the same time, they're going to pump stuff into the cell. So, does anyone here have a sump pump? Nobody? Oh, all right. Darn it. So, yeah, in some basements you have them. Was it working after the hurricane came through? Oh, you never lost power?
I guess it's working during the storm.
Okay. But yeah, here's the thing. Pumps of any kind need power to work. Same thing for these. They require ATP, right? And so we're going to talk about some of these pumps. One pump in particular is going to take up almost 66% of the total energy expenditure of your cell. All right, we'll get there. All right, so these pumps, and this is important, these pumps, you need to memorize this, will maintain a concentration gradient. Y'all remember what a concentration gradient is, right? Where you have more of something in an area and less of that same something in another area, okay? We saw it with diffusion. We saw that there was more oxygen outside the cell than inside the cell. And so oxygen wanted to go inside the cell. And we saw that there was more carbon dioxide inside the cell and less outside the cell, so carbon dioxide would want to leave. So this is what we're talking about, a concentration gradient. So when we talk about our pumps, we have these transport membrane proteins that maintain a concentration gradient by moving substances, and this is important, against their concentration gradient. Against their concentration gradient. I need to tell you this right now before I forget it, and I'll get into it not in this class, I mean, not tonight, but in a later class. Banana floating in the ocean. I know. Just imagine that. Think of it in your head. Think of a banana floating in the ocean. I'll explain what that all means later on. Right? When you think of a banana, what is a banana high in? Potassium. Good. Okay. So think of your banana as your neuron. What's significant about the ocean? Yes, it's water and whatnot, but salty. And what is salt? You remember the molecular formula for salt? Yep, sodium and chloride. Yep, that's right. Okay. So just keep that in mind, and I'll elaborate that more so, but I just wanted to give you something to chew on here. So we have two types of pumps that we're going to talk about. We have our sodium potassium ion pumps, and then we have our calcium pumps. And these are located in the membrane of our neurons. And we'll talk about the specific area as to where they are located, all right? But just keep that in mind. So these pumps are pumping, but they're going to be pumping things against their concentration gradient, which means if you have more of something outside the cell and less of it inside the cell, these pumps are going to keep pumping whatever it is that they're pumping to the area where there's higher concentration for it, right? So that means they're going to go against the concentration gradient here. So the other type of transport protein that we'll be talking about are the channels, right? And I want you to think of channels as holes or pores in the membrane, in the plasma membrane, that are going to allow ions. What's an ion? Charged molecule, right? It's either positively charged or negatively charged, okay? Like calcium, sodium, potassium, chloride. All of those are ions, right? So these special transport proteins are going to allow these ions to move down their concentration gradient, which can mean it can either go into the cell or out of the cell. All right, so we saw that pumps pump things against their concentration gradient, and they require energy, but channels are going to allow the movement of ions, right, down their concentration gradient. So it depends on where there's a higher concentration. So that means it can either move stuff into the cell or out of the cell. So channels that we're going to learn about are going to be specific to one type of ion. Right? So you're going to have transport proteins or channels that we call voltage-gated calcium channels or voltage-gated sodium channels. So the name tells you what ion can diffuse through that channel. So if I have a voltage-gated sodium channel, knowing what I just talked about here, can calcium go through my voltage-gated sodium channel? No, that's right, because it's specific, right? Our voltage-gated sodium channel is specific for sodium only. Calcium, potassium, none of that other stuff can go in there. So there's a couple different types of channels that we're going to talk about. All right, the first one is the 7-Eleven channel, okay? Those are what we call leak channels. They're open 24/7, never ever closed. They're not gated, right? They are not gated. So we'll see those channels for specific ions. So we'll have what we call sodium leak channels, potassium leak channels, which means all this stuff that we're going to learn in this chapter, all these processes that I'm going to go over with you, these channels are always leaking stuff. They're never closed. So when I talk about how we generate an action potential and how we do this and that, these channels are doing one thing only, and they're just leaking stuff into the cell and out of the cell, depending on what the concentration gradient is. All right, the next channel is called a chemically gated channel. Two things here I want to draw your attention to: chemically and gated. Let's do the easy one first. Gated. There's a gate on this channel. So that means that this channel can be in one of two positions. Either it's open or closed. If the gate's closed, nothing's going through the channel. If the gate's open, stuff is moving through the channel. Chemically means that we need a neurotransmitter to open this gate. So when you see chemically gated, you should be thinking, all right, a neurotransmitter needs to bind onto this gated channel to open it up. So the neurotransmitter is like the key that opens up that channel. And again, it's specific for one type of ion. So normally we name it for whatever ion. So we'll say, all right, we've got a sodium chemically gated channel. And so you would know, oh, all right, so I need a neurotransmitter to open up this channel, and then sodium can flow through that channel. All right, we name it that way. All right, the other one is called voltage gated channel. Same thing though. We got a gate on this one, so it's either open or closed. Normally they're closed. And then now in order to open up my gate, I need some sort of electrical charge, a change in what we call the membrane potential. And we'll get to that. We'll get to that. And the same thing happens. It's ion specific. So if we have a voltage gated potassium channel, what ion goes through that channel? Potassium. Yeah. Right. So voltage gated sodium channels, right, are going to only allow sodium. Now, speaking of voltage gated sodium channels, these are special gates or special channels that I'm going to talk to you about. All right. So it's actually good because we're going into that. So our voltage gated channels actually have two types of gates. I want to make a note of that. You have activation gates and inactivation gates. All right, the other ones that we've been talking about, the chemically gated and just the regular normal voltage gated channels, they have one gate. But voltage gated sodium channels have two: activation and inactivation gates. So let's talk about those. All right. It has two gates, but it has three states. Right, so these channels have two gates, an activation gate and an inactivation gate, but yet they have three states, three phases, whatever you want to call it. We're going to call them states. So you have the resting state, the activation state, and the inactivation state. So when you're at when this channel is at the resting state, right, nothing's happening, it's just relaxing. The activation gate is closed. I'm going to show you a picture of that here real quick, and then we'll come right back. All right, that's what we're seeing here. So you have your you got your resting state here. So it's closed. This is the activation gate down here at the bottom. It's that little swing arm that you can see here. And then you have another gate. You can kind of see it here. You can see it a little bit better, which is called the inactivation gate. See it a little bit better over here. All right, so the inactivation gate is going to be like these two little swellings. So when the inactivation gate is open, the swellings are like deflated. And then when we stimulate or activate the inactivation gates, they'll just kind of pinch together. Those two swellings will swell up and pinch together and close off the channel here. So in the resting state here, the activation gate is closed, the inactivation gate is open. When either gate is closed, nothing can get through. See that? Notice on this drawing here, we put in on the outside of the plasma membrane a positive charge. And then down on the inside of the cell, we have a negative charge. I'll discuss what that is, but this is the membrane potential. So for right now, in the resting state, the outside of the cell is positive, the inside of the cell is negative. We'll get into what all that means, but for right now, just memorize that. All right, outside is positive, inside is negative at the resting state. So, resting state, activation gate is closed, inactivation gate is open. That means no sodium is able to get through, because one of the two gates is closed. So sodium is denied. So now what we're going to do is we're going to stimulate this channel and open up the activation gate. So we call that the activation state. So when we change that, remember that electrical charge that I was telling you about? The positives on the outside, the negatives on the inside? When we change that, when we manipulate that, all right, we're going to change the voltage here. That's going to trigger the activation gate to open up. Well, the inactivation gate is still open. It's not changing. It was always open at this point. So now we got both gates open. Guess what happens? Not calcium. Sodium is able to move through the channel. Here. Activation gate is open. The inactivation gate has remained open. So sodium will start to enter into the cell. Now look. 1, 2, 3, 4, 5, 1. Where's there more sodium? Outside the cell or inside the cell? Outside. It's going to move down its concentration gradient. You don't believe me? Look here. Protein pores in the membrane that allow ions to move down their concentration gradient. So that's what we're seeing here in this picture. More sodium outside, less inside, so sodium starts to move into the cell until we reach equilibrium. So that's the activation state. Activation gate is open, inactivation gate is open, sodium can move through. What triggered the opening of the activation gate? We changed the positive charge on the outside to negative. We just flip-flopped it. It was positive outside, negative inside. Now we're just going to flip-flop it. Now it's negative outside and positive inside. So that electrical change, that voltage change, triggered the opening of the activation gate, allows sodium to enter in. The last state here is called the inactivation state. And what happens here is the inactivation gate closes, but the activation gate stays open. That little swing arm stays open. This happens, it's very brief, very, very short amount of time. And since one of the two gates is closed, that means tough luck, sodium, right? You cannot move across the plasma membrane. So no more sodium's getting in. As soon as this happens, we get into the inactivation state. I'll come right back to this slide. As soon as that happens, right here you can see the inactivation gate is closed. It's pinch closed. Activation gate still open. No more sodium can enter in. This happens for such a short period of time, right? It'll briefly occur, and then we quickly revert back to the resting state here. Because what will happen is the activation gate will close and the inactivation gate will open up. But again, right, it's blocked, so no ions can enter through there. So those are your three states. This is very important because we're going to talk about another concept here, all right, later on in this chapter that has to do with this unique, I should say, configuration of a voltage-gated sodium channel. Right, so just kind of keep that in mind, the different activation, not I should say the different activation gates, but the different types of gates that are on this type of transport protein. All right, then we have our last type of gates that I want to talk to you about, or gated channels I should talk to you about, are what we call modality gated channels. And again, most of these gated channels, they're normally going to be closed, and some sort of stimulus is going to trigger the opening of those channels. We just saw in a voltage gated channel, it was a change in the voltage. In the chemically gated channel, a neurotransmitter was needed. Modality gated channels, right? We need, all right, something other than, well, I just discussed with you, a chemical or a voltage change. It can be, and we'll talk more about this in chapter 16, change in temperature. You can apply some pressure. Remember those Merkel cells that we talked about that were in the skin, right? And if you press on a Merkel cell, you're going to deform the plasma membrane, and that pressure and that deformation of the plasma membrane as you press on it, it's going to cause it to release neurotransmitters that will stimulate a receptor. So you can have any of these stimuli, a change in temperature, some pressure, or even light exposure, like we'll see in photoreceptors in the eye, right? All of these will have some sort of effect on the plasma membrane here. And then that effect will then open up that modality gated channel, and then ions can move across. We'll get into more of that in chapter 16 when we talk about your special senses like sight and smell. All right, so where are these pumps and channels? Let me see something real quick here. All right. Well, in your book, it actually shows the whole neuron here. I'm going to go through this with you. All right, but you're going to see, all right, these pumps and channels, all right, certain pumps and channels will be in various locations, and then others will be all over the place. Like, for example, you've got your sodium potassium ion pump, and then we have our sodium and our potassium leak channels. These pumps and channels are located in the entire neuron. They're located here in the dendrites, in the cell body, down in the axon, and even right down here, right in the telodendron. So they're on the entirety of the neuron. So there's some other ones that are in specific places, and we'll talk about those. Let me just, I know I skipped here. Whoops, there we go. All right, so for the entire neuron, you will see these these two leak channels and our sodium potassium ion pumps. So those three types of transport proteins are responsible. This is an important concept, so memorize this. Those three things are responsible for maintaining the resting membrane potential. And what's the resting membrane potential? This, positive outside, negative inside. There's a better definition for that, but I don't want to overwhelm you right now. You guys just took a test, right? So just keep in mind, those leak channels and those pumps make it that when a neuron is not doing anything, like when you're sleeping tonight and you're not moving your arms and your legs, all right? The nerves that affect your arms, the muscles in your arms and legs, they're not really going to be doing anything. So they're going to be more like in the resting state. So when they're in the resting state, that means it's positive outside and negative inside. So nothing's happening. So why is that? It's because of these three structures here, right? The sodium potassium ion pump is going to be pumping sodium out of the cell while it's pumping potassium into the cell. And then the sodium leak channel is going to allow sodium to leak into the cell while potassium leaks out of the cell. Again, we'll get into this detail here, right, in a little bit. So keep that in mind. But the important concept that I want you to understand here is when I go over it later on, I don't want to have to revisit this because I want you to know it. All right. So those three things, sodium potassium ion pump, the sodium leak channel, and the potassium leak channel, maintain the resting membrane potential, which is this configuration: positive outside, negative inside. Good on that? I'll get into a better definition as to what resting membrane potential is. All right, so when we're looking at our neuron, when we're looking at the neuron, right, there's a couple different areas or regions that I want to talk about. And so there's 4 of them. So you have what's known as the receptive segment, the initial segment, the conductive segment, and the transmissive segment. All of these describe the functional segments. What's that mean? The physiology. So functional segments means the physiology segments of the neuron. Now, anatomically, if we were talking about these segments, we would refer to them as, like, for example, the receptive segment is made up of the dendrites and the cell body. That would be this here. There's the cell body and all the dendrites. So that's the receptive segment. And then you have the axon hillock. We refer to that as the initial segments because we're going to initiate our action potential. And then we have our conductive segment that's going to transmit that nerve signal. That's the axon. And then we finally have here at the bottom where the synaptic knobs are, that's the transmissive segment because it is going to transmit, right, the neurotransmitter, right, from this neuron across the synaptic cleft to either another neuron or to an effector there. So that's what the receptive segment is. You should just memorize this. Dendrite and cell body. Initial segment is the axon hillock. The conductive segment is going to be the axon and any branches that come off. Remember the collaterals that we talked about? If it sends out a bunch of different branches all over the place, that's the conductive segment. And then finally, the transmissive segment is the synaptic knobs. We're going to run through this and then we'll call it a night. So the receptive segment is going to have chemically gated channels, and it varies because there could be several different types, but normally it'll be, all right, potassium, chloride, sodium, chemically gated channels. The initial segment is going to have voltage-gated sodium channels and voltage-gated potassium channels. The conductive segment has the same voltage-gated sodium channels and voltage-gated potassium channels. Finally, the transmissive segment has voltage-gated calcium channels, and this is new, calcium pumps. And there's a reason for this. We'll talk through it. So I'll show you the nice picture here in your book. Or I mean, not in the book, well, on my slideshow. All right, so keep in mind, in the entire neuron, sodium potassium ion pump, and then our sodium leak channels and potassium leak channels, they're found throughout the whole thing. In the receptive segment, here you can see your chemically gated, all right, cation channel. An example of that would be like sodium, chemically gated potassium channels, and then chemically gated chloride channels. And that's only in the receptive segment here. So in the dendrites and in the cell body. Notice that they're all chemically gated, which means a chemical of which will stimulate the opening of those gates. And what chemical would do that? What's the broad name that we give it? It begins with an N, ends with mitter. All right, yes, neurotransmitter. So the neurotransmitters are going to be that chemical that are going to stimulate the opening of these gates here. We get down here to the axon hillock, right, where the cell body starts to taper down into the axon. That's the initial segment, right? It has both of these voltage-gated channels here, sodium and potassium voltage-gated channels. Then we get to the conductive segment all along the axon, same thing. Voltage-gated sodium channels, voltage-gated potassium channels, all the way down till we get to the synaptic knobs. And in the synaptic knobs, in the plasma membrane there, we'll see voltage-gated calcium channels in our calcium ion pump. Check it out, though. This is another reason, specifically, this right here is why it's important that you have calcium in your body. Because if you don't have calcium in your body at all, this will never work. And we won't be able to transmit action potentials. We cannot make nerve signals there because we don't have calcium there. So, question for you. What is the difference between a chemically gated channel and a voltage gated channel in terms of how they function? So for a chemically gated channel, what do we need to do to open it up? We need a neurotransmitter, all right? That neurotransmitter will then open up, all right, that channel there. For a voltage gated channel, what do we need to open that channel up? Yeah, we're going to need some sort of change in the voltage there. So we're going to have something that has to do with the electrical charge. Remember, it was positive outside the cell, negative inside the cell. We need to have a change in that, something that's going to cause a change and make it negative outside and positive inside, which is what leads me to the next slide, which is a perfect slide to end on. This slide here, banana floating in the ocean. Take a look at it, all right? And when we're done, you will understand what I meant by banana floating in the ocean. All right, y'all were wonderful to sit through this with me after your test. All right, I know it's not easy to do.